PCR uses the enzyme DNA polymerase that directs the synthesis of DNA from deoxynucleotide substrate on a single-stranded DNA template. PCR can be performed in two basic steps. The first is the strand separation. The two strands of the parent DNA molecule are denatured by heating the solution to 95 Celsius for 15 seconds. The second step is the hybridization or annealing of the two oligonucleotide primers when the solution is cooled to 54 Celsius. The third one is the DNA synthesis or elongation, which occurs by heating the solution to 72 Celsius, which is the optimal temperature for the DNA polymerase.
Different kinds of PCR. In the multiplex PCR, several primer pairs to various target sequences are used to enable simultaneous analysis of one or more sequences of interest. This is useful for testing genetic mutations, DNA fingerprinting protocols, analysis of microsatellites, and SNPs.
Reverse transcriptase PCR. PCR is preceded by a reaction using the reverse transcriptase as an enzyme that converts RNA to DNA. It's useful when transcripts are in very low abundance and the amount of the starting RNA is fairly limited, such as from a protoplast transaction. Real-time PCR. A fluorescent reporter molecule is used to monitor the PCR as it progresses, and at the same time, a relatively small amount of PCR product, DNA, cDNA, or RNA, can be quantified. It's useful for quantitative mRNA expression studies, DNA copy number measurements in genomic or viral DNAs, allelic discrimination assays or SNP genotyping, verification of microarray results, drug therapy efficacy, and DNA damage measurement.
qRT-PCR. In this method, RNA is first transcribed into complementary DNA by reverse transcriptase from total RNA or messenger RNA. The cDNA is then used as the template for the quantitative PCR reaction. Its applications go to gene expression analysis, RNA validation, microarray validation, pathogen detection, genetic testing, and disease research.
Nested PCR. It involves two sets of primers used in two successive runs of the polymerase chain reaction, the second set intended to amplify a secondary target within the first run product. It's used to increase the specificity of the DNA amplification and for clinical applications.
Hot start PCR. The method involves withholding one of the critical components of the PCR from the reaction until the temperature in the first cycle rises above the annealing temperature. This can be performed manually by heating the reaction components to the DNA melting temperature, or by using a physical barrier like wax to segregate the key reaction components. It's useful because it reduces non-specific amplification and offers the convenience of reaction setup at room temperature.
Colony PCR. Small quantities of bacterial cells from bacterial colonies are added to the PCR mix. An initial extended annealing period or a shortened denaturing step at 100 Celsius is used to release the DNA. It's useful when screening positive transfer DNA vector constructs or desired recombinant clones. Polymerase cycling assembly. Long DNA molecules are created from long oligonucleotides with short overlapping segments. Primers with an overlap are used to create products that can then be used as a template to generate the long DNA molecules. This is useful to assemble two gene-sized pieces of DNA into one piece for easier cloning or fusion genes part. Droplet digital PCR. A sample is fractioned into 20,000 droplets, and PCR amplification of the template molecules occurs in each individual droplet. Its applications go from liquid biopsy to copy number variation, rare sequence detection, gene expression and miRNA analysis, single-cell analysis, pathogen detection, and next-generation sequences library analysis. To perform PCR amplification, you need to combine all the components required for DNA replication. These are as follows. DNA template. The template carries the DNA segment or target you wish to amplify. Primers. A primer is a short, single-stranded piece of DNA that anneals to its complementary sequence on the template. A pair of primers will bind to either side of the target DNA segment, providing initiation sites for DNA synthesis. The two primers used in a PCR are often designated by the terms forward and reverse. DNA polymerase. This is the enzyme used to synthesize new strands of DNA. The DNA polymerase adds nucleotides onto the end of an anise primate. The other bases are complementary to the template strain. Magnesium. Thermostable polymerase requires the presence of magnesium to act as a cofactor during the reaction process. Its role is similar to that of a catalyst. The magnesium is not actually consumed in the reaction, but the reaction cannot proceed without the presence of the magnesium. It is usually supplied to a PCR amplification in the form of magnesium chloride. DNTP stands for deoxynucleoside triphosphate. These are 4 nucleotides used for DNA polymerase to extend an allyl primer. Buffer. A buffer is a solution that resists change in pH. It is used to ensure the DNA polymerase an environment in which it will have maximum activity. One of the basic components of PCR is the primer design. Here's an example. The CoA gene from E. coli encodes for phosphopanthein adenyl transferase, an enzyme involved in the biosynthesis of coenzyme A. Here is the CoA gene, the sequence. We decided to clone the gene into an expression vector using the restriction enzyme NCO1, 5'N, and BAMH1 in the 3' end. Show the design of both primers. The forward primer has a 5' extension of CATG and NCO1 restriction site with the CCATGG and the start codon ATG. They overlap with the 5' end of the CoA gene, starting with the ATG start codon, and an estimated temperature of 62 Celsius and a GC content of 48%. The reverse primer has no C-terminal tag, and so that we add a stop codon of TAG is a natural stop codon for PPAT. So it has a 5' extension of CG, the BAMH1 restriction sites as GGATCC, the complement of the stop codon CTA, the overlap with the strand complement to the 3' end of the CoA gene, and an estimated temperature of 60 Celsius and a GC content of 60%. According to the primers used in this lab practice, the ZFX and ZFY can be used for gender information, which particularly when it's combined with the mitochondrial DNA analysis can serve to distinguish biological evidence from 2 people who share the same DNA types but differ by sex. When typing sexual assault evidence, gender information can serve as a confirmation that the sperm fraction extracted from the swabs and stains actually contains the male DNA, and also as an indicator of the amount of the male DNA present in the known sperm fraction.
Sexing by PCR of female and female DNA's electrophoresis was carried out in 0.8 agar gel with TAE buffer. The bands were visualized under ultraviolet transilluminator. Lanes 9 have 1 kilobase plus, lane 10 had 3 microliters of DNA sample, lane 11 had 3 microliters of negative control that was water, and lane 12 had 3 microliters positive control that was PCR products. The agar gel was strained using gel red. In the case of the primer forward, we have a delta G of minus 0.98. At this energy is required for the breaking the structure. The homodimer has a delta G of minus 5.02. and the heterodimer has a delta G of minus 5.25. In the case of the primer reverse, it has a delta G of minus 0.28. It is the energy required to break in the structure. And the homodimer has a delta of minus 549 delta G. In the next chart, we have the specification of the primers. In the first column, we have the forward primer sequence that was 5' to 3'. In the second, we have the reverse sequence primer that is 5' to 3'. And in the third, we have the temperature of melting of each primer. Fourth, we have 58.3 degrees Celsius, and for the primer reverse, we have 55.1 degrees Celsius. We conclude that if they are not very stable and can be used, that don't significantly affect the process because they do not form the secondary structures. In the lane 4, we have a PCR product degradation that is in color pink. and in color green we have PCR product. This is in the band 5000 and 3000. And in the color blue we have the NTPs and primers. This is running more than the marker. We have 0.8 agarose gel on lanes, 1 kilobase in vitro gene marker and was 5 microliters, 2 genomic DNA sample 5 microliters plus 2 microliters of loading buffer, 3 negative control that was molecular biology water grade that was 5 microliters plus 2 microliters of loading buffer. 4 we have positive control of PCR products of 447 pairs of base that was 5 microliters plus 2 microliters of loading buffer.
When performing a PCR, one has to take care of several things, including per-PCR activities, which include protocols and equipment necessary for nucleic acids extraction, sterile and contaminant-free reagents, and working space, as well as good care during sampling are critical for a PCR reaction. Also, when preparing the PCR reaction, the amount of polymerase, DNA TPs, buffers, and primers should be added according to the concentration of the sample to be amplified. Before starting the PCR reaction, parameters such as time, temperature, and number of cycles need to be established according to the enzyme used and the primers added. If a PCR cycle is set without considering this, it must likely to fail. Most of the times, an unsuccessful PCR resides in the quality of the sample, which may be affected by the extraction protocol performed, contamination by DNA or salts, among other things. According to Laura Espinosa from the National Institute of Ecology and Climate Change, if you're to perform several PCR reactions, it is highly recommended to use an extraction kit from a reliable vendor. That way, it is mostly guaranteed to have a better outcome. On consequence, we agree that above all steps during the preparation of performance of PCR, it is critical to have excellent pipetting skills in order to avoid contamination and/or loss of samples. Also, it is important to know the working parameters of the enzyme used and the proper concentration of reagents for a PCR reaction. About the results obtained in this practice, the most significant would be the fact that the DNA sample is not observed on line 2. We attribute this event to the fact that during DNA preparations, the sample was quite diluted with water, and maybe that's why there is no PCR product. It is remarkable that, as expected, on lane 3 there is no sign of DNA contamination, therefore it can be assumed that the protocol was successfully followed. As for lane 4, as expected, the PCR product is observed, with little degradation and a possible cell contamination. At the very bottom of the agarose gel, in lanes 2 to 4, it can be observed 2 marks, which we attribute to the existence of unused DMTPs and primers during the PCR reaction. This may be possibly due to the high concentration of DMTPs, but this is not a determinant factor for the results, because if more DMTPs are added, this may kidnap magnesium and inactivate the polymerase. Therefore, the zero obtention of DNA on lane 2. But the same would have happened on lane 4, but it didn't.
Polymerase chain reaction (PCR) is one of the most used methods for amplification of sequence of interest. In this work, such method was used to a genetic easy determination of gender, including a co-amplification of zeta if X and zeta of J target. In order to obtain a successful outcome, it is necessary to have an appropriate procedure, which starts from the pipetting skills, preparation of the reagents, as well as sample and the correct handling of these. It is important to remark that the extraction of nucleic acid was a critical point at which contamination can occur with proteins, DNases, enzymes which are responsible for the degradation of genetic material, and other reagents that can alter the results and therefore report basis conclusion. Also, when performing PCR, number of setting cycles of extremely important because the change of temperature and excess of time can lead to the inactivation of the polymerase. Lack of PCR reagent or even amplification of unspecific reagents, thus drawing basis results.